Hello, Assalamu alaikum. In this video, we will understand about the thermogenesis via a vitamin B1. Vitamin B1 is known as the thiamine. The availability of abundant thiamine determines the efficiency of thermogenic activity in human neck area derived adipocytes. But in the research, the human deep neck area, but a lot of area involved, but study shows that uh, less effect. This is the deep neck cell, which that activation due to the vitamin B1. The DNA will activate to produce certain genes for the biogenesis of the mitochondria as well as the UCP production. The UCP1 gene activation to produce more UCP will enter into the mitochondrial inner membrane, and this is the adipose tissue, while on the other hand a lot more other factors transcribe to the biogenesis of mitochondria. Exercise, fasting and diet will also lead to increase with the vitamin B1. The increase the mitochondria is known as the mitochondrial biogenesis or mitogenesis, lead to more UCP1 and more heat production. This is the BG. More BG adipose tissue contain a moderate amount of mitochondria. More exercise and cold exposure will lead to increase the brown adipose tissue in the presence of the thiamine. Let's understand about the basically is the cellular pathway and how it can trigger the DNA to produce more UCP1. Anyhow, this is the channel THTR1 and 2 involved for the thiamine or vitamin B1 entry into the cytoplasm from the extracellular matrix into the intracellular matrix. This thiamine phosphokinase enzyme, which that produce the thiamine pyrophosphate. This thiamine pyrophosphate, and on the other hand, the sympathetic nervous system activation due to the cold exposure lead to adrenergic neuron to produce adrenaline through adrenergic stimuli. This adrenaline will bind with the G-coupled protein receptor to activate the adenylate cyclase enzyme. This adenylate cyclase enzyme will lead to activate the AMPK into the cyclic AMP due to the adenylyl cyclase enzyme. So it will lead to become a cyclic AMP from the simple AMP. The cyclic AMP will trigger the basically is the cascade of phosphorylation. We will understanding with the missing this we will understand shortly. Will lead to activate the transcription factor and activate the UCP1 gene and the PGC1 alpha for the biogenesis of mitochondria, because the PGC1 alpha will lead to increase the TFAM. This TFAM is protein, but on the other hand, the glucose through GLUT1 and 4 channel enter to glycolysis to produce pyruvate. This pyruvate is the precursor for the production of ATP, while also the UCP function for the production of heat. This UCP1 and PGC1 alpha will enter into the mitochondria, and biogenesis of the mitochondria and UCP1 when rise. In this way, the basically thiamine pyrophosphate also enter to bind with the pyruvate dehydrogenase enzyme. This pyruvate dehydrogenase enzyme will convert when enter the pyruvate into the mitochondria to acetyl-CoA. This acetyl-CoA basically will enter into the tricarboxylic acid cycle in the inner mitochondrial matrix. When inner mitochondrial matrix to produce more FADH2 and NADH2. This FADH2 and NADH2 in the is shown in the figure, which that will pump out the proton in the outer mitochondrial matrix, and in this way the proton gradient will arise, and in this way the proton will move into the inner mitochondrial matrix through UCP to produce heat.